welcome to the Talking Crop podcast. My name is Catherine Seabrook, and I'm a commercial agriculture educator with University of Illinois Extension, serving Daviess, Stephenson, and Winnebago counties. Talking Crop is a row crop production podcast with episodes occurring every other week during the growing season between May and September. In each episode, I bring on a guest speaker to discuss topics related to their areas of expertise. In today's episode, I'm joined by Dr. Patrick Tranel, professor of weed molecular biology and genomics in the Department of Crop Sciences at University of Illinois. We discussed his research in weed genomics and herbicide resistance, as well as the implications of metabolic resistance as it relates to the future of weed control. In the episode description, you'll find a link to a webinar that Dr. Tranel did with the Illinois Soybean Association to discuss his lab's characterization of glufosinate resistance in water hemp, as well as a link to the paper that they published to document this novel resistance case. For the next episode of Talking Crop, Dr. Allison Robertson, professor of plant pathology and microbiology and extension field crops pathologist with Iowa State University, joins me to discuss field crop disease development, forecasting, and fungicide return on investment. That episode will air on Wednesday, June 10th. And now, please enjoy this episode of Talking Crop, The Future of Herbicide Resistance with Patrick Tranel. Hello, Dr. Tranel, and welcome to the Talking Crop podcast. Thank you so much for joining me today.
Hello, Catherine. Thank you for having me.
Absolutely. So I would first like to start off by having you introduce yourself to those listening. So just who you are and mainly what you do.
Sure. So my name again is Patrick Tranel. I'm a professor here at the University of Illinois. I joined the faculty here at this campus way back in 1997. So I've been here pushing on 30 years here pretty soon. So obviously this is a great place to be. During that period of time, my research is focused on weed science. I'm also involved in teaching both undergraduate, graduate courses in weed science. I also train students for a weeds contest. Believe it or not, there is such a thing. In terms of my research, my focus has really been on herbicide resistance. Some of my previous training before I came here was actually in molecular biology. And so I was hired at the University of Illinois specifically because it was recognized at that time that there was not a lot of molecular biology research being done in weed science, and there were some opportunities, there were some questions that needed to be addressed using a molecular approach. And so that was why I came here, and that's kind of what I've been doing ever since. Of course, in the last few years, we've transitioned from not doing just molecular biology, but now doing genomics. And so my lab has been involved with a lot of genomic sequencing and then using these genomes to address questions in ways we couldn't address them ever before. So, for example, we have now a very, very high quality water hemp genome that we can use for mapping of herbicide resistance traits, for example. So that's kind of a quick overview of what I do.
Thank you. And a little bit of history on my end. Dr. Tranel was my grad school advisor. I think since I left, you guys have shifted a little bit towards some other focuses, but we'll get to that in a little bit.
I also remember that you competed in the weeds contest at least once, maybe twice. I don't remember.
I didn't want to bring it up because I didn't do very well. My team was not as good as the teams that you have had recently, I will say. But yes, I have participated in the weeds contest. I still have the t-shirt, so I do have that little piece of history with me still. So, Pat, producers, they might be more interested in more applied weed science research. I'm on the other end now. I'm in extension. So I work with producers who, again, they want more of that applied information. Or in essence, what herbicides should I use to control my weeds? Should I tank mix? Should I rotate? Things of that nature. And then what you do is kind of, in a sense, kind of looking under the hood of what's happening in weeds when it comes to herbicide resistance. So it's a very different approach. So how would you explain, why the research that you do, why is that still pertinent to producers?
Yeah, before I get into that, I just want to do a shout out to my colleague here, Aaron Hager, who is that applied person that you're talking about, who answers those more specific applied questions. And Aaron's been here as long as I've been here, and we've had a great relationship. And I think we really sort of exemplify the land grant mission, where he's the eyes out in the field. He's talking to farmers. He sees their issues that they're dealing with. he addresses them as he can, but sometimes, it requires science that he's not able to do. And so he brings those questions to me. I do the more basic research, come up with solutions, pass that information to Aaron, who then takes it back out to the farmer. So we have this nice sort of exchange between the farmer, or from the farmer and back to the farmer through this applied basic research. Why does what I do matter to the farmers? I guess one place to start is herbicide resistance is an evolutionary process. It's evolution in action. It doesn't just magically show up in your field all of a sudden. Those weeds evolve. And evolution is a very scientific process. It's actually a fairly predictable process, really, if you understand some of the parameters that are involved. And so, as farmers, we want our herbicides to be sustainable. We don't want the weeds to evolve resistance. Unfortunately, evolution happens, and it's a very strong driving force. But essentially, what we would like our farmers to be able to do is to slow down that evolutionary process. And in order to slow down that evolutionary process, we have to understand the details of how it's working. Okay. And so, you know, the research that I do is typically not going to give a farmer an answer tomorrow. It's more long term, but it's helping those farmers better understand, why should we rotate herbicides? Why should we mix herbicides? In fact, some of the research that I was involved with was pivotal in changing the focus from rotating herbicides to mixing herbicides. And so there's a very specific example. A long-term management recommendation was simply rotate herbicides. Use group 1 this year, group 9 next year. Okay. Now our recommendation is you really need to use group 1 and group 9 simultaneously this year. You have to mix those effective modes of action, because some of the research that we did helped us determine that simply rotating herbicides was not slowing down evolution of target site resistance that we were looking at then. Of course, now we're in a different ballgame of metabolic resistance. And so managing target site resistance is quite a bit different than managing metabolic resistance. But we wouldn't know we had metabolic resistance if there weren't people like me that was doing that research to determine what those resistance mechanisms are. I guess another thing that I'll point out, one way that the basic research that I did had a very applied value was that, way back when glyphosate Roundup resistance was just kind of becoming a thing, we were starting to see a lot of water hemp resistance to Roundup. That was back probably 15 plus years ago now. Farmers were wondering, well, if I got Roundup resistance, what could I spray? And back in the day for soybeans, post water hemp control in soybeans, you really only had two options. The group 2, the ALS inhibitors, or the group 14, the PPO inhibitors. And so, we had ALS resistance in our water hemp at the time. We were starting to get some PPO resistance in our water hemp at the time. And so what a farmer wanted to know, if they sprayed Roundup and they didn't get good control, they wanted to know, A, is it resistant to Roundup, or did I just have a bad year that year? It didn't work for whatever reason. And if it is resistant to Roundup, is it also resistant to the PPO chemistry and the ALS chemistry? And so because we knew the mechanisms of resistance to all three of those, glyphosate, PPO, and ALS, we were able to develop molecular assays to screen for those resistance. And so we did that, and we actually transitioned that. The Illinois Soybean Association funded some of that research, and we did that as a free service to farmers initially. It was paid for by the Illinois Soybean Association, where they could simply send us water hemp samples, and within a few days, we could tell them the resistance profile that they had to those three herbicides. And so then we did it for a year or two, and then we transitioned it to the plant clinic here on the campus. And I believe they still run that assay. Obviously, there's not that much interest in glyphosate resistant water hemp anymore. Farmers know they have it, but that test is available. And so that was a very practical outcome that was completely dependent on the basic research that my group and others did.
So when it comes to resistance, I sometimes wonder with producers, if they don't have resistance or don't know or think that they have resistance, it might seem like this sort of, out there issue that will never affect them. Can you kind of paint a picture of the state of herbicide resistance today? I mean, is it common? And then B, has it remained steady, or is it increasing in frequency?
So obviously here in Illinois, water hemp is the biggest problem. So most of my, discussion will focus on that weed. I don't think there's any farmers in Illinois that do not have resistant water hemp. Okay. I mean, it's hard to find a population of water hemp that is sensitive to every herbicide. Okay. For example, resistance to ALS inhibiting herbicides group 2 has been common in our state for years, maybe for 2 decades, certainly for at least 1 decade, where ALS inhibiting herbicides, basically we stopped recommending them for control of water hemp. So we've lost what most farmers don't realize how great the group 2 herbicides were at one point. They were fantastic herbicides, but we've lost that tool for water hemp management for all practical purposes. So that's one example. There's glyphosate resistance is very common in water hemp. I would say it's harder to find a glyphosate sensitive population than it is to find a glyphosate resistant one. There's still some glyphosate sensitive populations, but they're becoming fewer and fewer. PPO resistance is the same thing. I estimate that for glyphosate, ALS, PPO resistance, maybe even atrazine resistance as well, I would say that's in the majority of water hemp, so over 50%. And one thing to keep in mind is if you have a population, so we're talking a field, you have a 100-acre field, and you have PPO resistance in that field. That doesn't mean that every single water hemp plant in that field is resistant to that herbicide. Maybe 10% are, maybe 80% are. I don't know. It's some range. So I think farmers should be aware of that. So, because of that, you might have resistance to PPO inhibitors in your field, but that doesn't necessarily mean that adding a PPO inhibitor to your tank mix has no value, because there might be a lot of individuals in that field of that same species, let alone other species, that are going to be controlled by adding that mixture or that tank mix to your herbicide. So that's something for farmers to keep in mind. It's not always the whole field. We're talking about individual plants in that field that express resistance. So those 3 resistances are very common, or 4, I guess: glyphosate, PPO, ALS, atrazine. We also have resistance to 2,4-D and dicamba. Those are not as common. I think we're probably going to see an uptick in 2,4-D resistance this year. I think growers have kind of started seeing some issues last year, and that's probably going to increase. What am I missing? Group 15, so soil applied, we've confirmed that in our state a few years ago now. Aaron's group has done some screening for that. That actually seems to be, more common than what we think. I don't know if you've had Aaron or anybody else talk about group 15, but that's, an interesting topic, because that's resistance to a pre-emergence product. And it's a lot different story, knowing whether you have resistance to a pre-emergence product versus a post-emergence product. HPPD resistance is quite common as well. That'd be the group 27. And then we've recently confirmed glufosinate resistant water hemp in our state as well. So if you think about herbicide options, post-emergence herbicide control of water hemp and corn or soybeans, is there a chemistry I didn't just name off? I mean, we basically have resistance, at least some resistance, to all available post-emergence herbicide, as well as, the group 15, a pre-emergence product. So it is a huge problem. And it's, how fast is it growing? It depends on the chemistry. Some of these have sort of leveled off by now, because you can only get so much. So, the frequency is only going to get so high. But I would certainly, glufosinate, 2,4-D, dicamba, HPPD, those are growing problems and probably will continue to grow.
And you mentioned how water hemp in Illinois has different populations have evolved resistance to all of these different sites of action that you've mentioned. And it's not to say, of course, that means then that every single population of water hemp in Illinois has evolved resistance to those. It's just that now we know that it is possible and probable in some cases if nothing is done to prevent that. Would you agree with that?
Yes, that's correct. And water hemp, one of its characteristics is it's dioecious, so plants are either male or female, which means a female in your field might receive pollen from a water hemp plant in your neighbor's field. And so if your neighbor has resistance, that resistance could move into your field as well. And so, and not only do we have resistance to these different herbicides, but because of that characteristic I just talked about of being outcrossing and being dioecious, is water hemp is very effective at stacking different resistance traits. And so a lot of times you will have a water hemp plant, a single plant that could have resistance to 2, 3, 4, 5, even 6 different herbicides all within one plant.
So you mentioned earlier that focus on target site resistance and how we've kind of shifted more towards what we call non-target site resistance, or maybe more commonly metabolic resistance. So can you explain briefly kind of the difference between the two?
So target site resistance, target site refers to the target of the herbicide. Okay, most herbicides work by binding a specific protein inside the plant, and they prevent that protein from doing its job. That's how the herbicide kills the plant. When we have target site resistance, what that means is that target, that protein, is changed such that it no longer recognizes the herbicide. The herbicide can't bind to it. And that change is usually a mutation in the gene that encodes that protein. It just changes the three-dimensional structure of the protein so the herbicide doesn't bind to it. That's target site resistance in a nutshell. And a key characteristic of that is that target site resistance only affects herbicides that have that same target. Makes sense, right? If a herbicide binds something else, it doesn't matter if the target site of another herbicide has changed. So it's very specific to that herbicide group, that herbicide group or that herbicide family, that class of herbicides. Metabolic resistance is one form of non-target site resistance, probably the most common form of non-target site resistance. And metabolic resistance is simply where the plant evolves the ability to metabolize the herbicide or detoxify the herbicide. So it breaks down that herbicide molecule into something that's no longer herbicidally active. Okay. And typically, most plants can metabolize most herbicides. It's really a race. It's how fast can it do it. When a herbicide is sprayed on a plant, the plant takes up that herbicide, takes up lots of molecules of those herbicides, that herbicide, and then it's a race. Do those herbicide molecules kill the plant first, or does the plant detoxify those molecules before it can kill the plant? The reason that you can spray atrazine on corn without corn dying is that corn has the ability to metabolize atrazine, and it can do it fast enough. Foxtail, a grass weed can't metabolize the atrazine fast enough, and the atrazine kills it. So as I said, most plants can metabolize most herbicides. What happens when you get metabolic resistance is over time, evolutionary time, that plant acquires the ability through evolution to metabolize that herbicide faster. And it metabolizes it now fast enough so the plant wins the race and it can detoxify the herbicide before the herbicide has a chance to kill the plant. And the challenge with metabolic resistance is that herbicides can be in different site of action groups, but they might be able to be metabolized by the same machinery, the same enzyme. Okay. A great example of this that farmers might be familiar with is enlist corn. Enlist corn was developed to metabolize 2,4-D. Well, it turns out that enzyme that can metabolize 2,4-D can also metabolize what we call the FOP herbicides. So those are a subset of the group 1 herbicides like quizalofop, okay? Quizalofop and 2,4-D, completely different herbicides, right? 2,4-D's broadleaf herbicide causes epinasty. The FOP herbicides are grass herbicides, cause slow death of the meristem. It doesn't matter. They have a part of their structure is similar enough to each other that it can be recognized by that same detoxifying enzyme. And so that's why they are resistant. That's why crops with the Enlist technology are resistant to both of those herbicides. It's due to a single metabolic mechanism. In the same way, we can have weeds that can acquire a single metabolic mechanism that can metabolize diverse herbicides, potentially spanning 2 or even more than 2 herbicide site of action groups.
And the thing with that makes it so difficult from a management standpoint then is us not being able to predict necessarily always which herbicides those plants that do have that resistance mechanism, we don't know which herbicides exactly that plant could be resistant to because it's metabolizing them, right?
That's exactly right. The only way we know with our current state of knowledge is, trial and error. Spray it and see if it works. Unfortunately, that's where we're at. That's a challenge, after you get resistance. It's also a challenge before you get resistance because for the same line of reasoning, we can't tell a farmer which herbicides they should be mixing and rotating to prevent that resistance evolution, because we might tell a grower to mix 2 different herbicides that have different site of action groups, but it might be possible for those 2 herbicides to be metabolized by a single mechanism. And so that it's a challenge at the front end of trying to delay resistance evolution, and it's a challenge at the other end once you get resistance, knowing what to do. And I think another challenge too is with metabolic resistance, there's exceptions to this, but in general, metabolic resistance is less black and white than target site resistance, in the sense that with ALS resistance, for example, target site ALS resistant, if you go out and spray water hemp population that's got target site ALS resistance with an ALS inhibiting herbicide, you won't even be able to tell you sprayed the herbicide. It would not affect the plants. Whereas we'd also have metabolic ALS resistance in our state. We don't worry about it too much because we got target site resistance. It doesn't matter if you got both. But the point is, with the metabolic resistant water hemp plants, if you spray them, they will look injured. And you might come out and scout your field a couple of days after your spray and think, hey, it looks good. I'm injuring my water hemp. But then they will win that race. They will continue to metabolize that herbicide, and then they will grow back. And so it's a little hard to see. It's a little hard to know whether you have resistance or not, or maybe you just got unlucky and you had a rain event 24 hours after you sprayed or 12 hours after you sprayed, and so you blame it on that. And maybe it was due to the weather conditions when you sprayed or after you sprayed, or maybe it was due to resistance. It's harder to know when you have metabolic resistance.
And you mentioned how we currently don't know how to predict what herbicides then to use if you have a metabolic resistance issue in a population. Is there research ongoing to try and come up with that predictive power?
Yes, there is. And I mentioned at the beginning here, we're talking about how we're using genomics to map herbicide resistance, for example. The other challenge with metabolic resistance, it's harder to determine what the mechanism is in the lab, right? Target site resistance, the target site, you go look specifically at the target site. Metabolic resistance, there's lots of different players that can be involved with metabolic resistance. When I say players, I mean proteins, and proteins are encoded by genes. So a bunch of different genes that might be conferring resistance. And so we don't know what those are. We really have no idea. You have to kind of do a fishing trip. And one way to do a fishing trip is to use genetic mapping, which, as I mentioned, now that we have a water hemp genome, we can do that. And so we have efforts in my lab, and other labs are doing this too, where they're using these modern genomic approaches to try and understand and identify the specific mechanisms or the specific enzymes that are involved with metabolic resistance. And then when we can find those enzymes, then we can use other approaches to determine which other herbicides might those enzymes be able to metabolize. And so the short answer to your question is yes. And I'm sure most of your listeners are familiar with the color-coded charts that we use that group herbicides by site of action. In the future, it might take us a while, but in the future, I'm hopeful that we will have a similar color-coded chart, which will group herbicides not by their site of action, but it'll group herbicides by how they're metabolized. And so you can say, okay, these two herbicides have different sites of action, but they can be metabolized by the same enzyme. And so we shouldn't be mixing those herbicides. So, that's going to be in the future, but there is progress at least being made towards that goal.
Yeah, that's relieving, and it'll be interesting in the future to have that resource available. I'm sure you're very familiar with all the different kind of like eras of weed control, and to be entering into that one will be something. Of course, it's a reflection or a result of not so great things, but to be able to come up with these tools for producers is really significant regardless.
Yeah, I could imagine a really nice app where you'd enter your weed species, and it would give you tell you what to spray based not only on what herbicides are effective on those weeds, but also do the herbicides have different sites of action? Are they metabolized by different enzymes? So it could put all that together. And of course, with AI now, this is not, this is not that far in the future, right?
And it's interesting too, because with the increase in herbicide resistance and with the increase of, I'll say it's complexity, there's also, I've noticed in the weed science research world, this shift towards looking at weed control, not using herbicides, but using other alternate methods like cultural methods as well. I know, like going to the different conferences over the past few years, there's been this huge influx of research on like cover crops for weed control too. So, in addition to that potential tool for the future and this more and more research coming out about these cultural control mechanisms, hopefully we'll start to see maybe a decrease in resistance issues. Fingers crossed. I know it's pie in the sky, but
one of the challenges, and I know this, from a farmer's perspective, you can use a cover crop and that will reduce your need for a herbicide, but typically it's not going to eliminate the need for the herbicide.
right.
And so if you got to spray the herbicide anyway, why spend the time doing the cover crop? I mean, I shouldn't be putting that into farmers' minds, but I'm sure they already have that in their mind, right? I mean, that's sort of their rationale. It's unfortunate, but it's necessity is the mother of invention, right? And
Yeah.
as long as farmers can get away with just spraying herbicides, I think they're going to continue to do so. Um, but I do think that, and I've been quoted in a magazine, farm magazine, a few years ago, that this metabolic resistance really is the beginning of the end of chemical weed control. Okay, the beginning of the end. And I'm not saying it's the end; it's not over yet.
Right.
And kind of an analogy I used with this was, back around 2010 when we started getting glyphosate-resistant water hemp, that was the beginning of the end of glyphosate. Okay, we still use a lot of glyphosate,
Hmm.
but back in 2005, people were using only glyphosate. That's all they needed. Now you can't use just glyphosate. You have to do something else. I think it's going to be the same way with chemical weed control. We're at the beginning of this era now where we're going to start seeing fields where you can't just use chemicals. You have to do cover crops or robots or something to supplement the herbicides.
And the thing with cover crops too that I always like to frame it from the weed control perspective with them is that vast majority of the time, I can almost guarantee you that a producer is not planting cover crops for weed control. I always like to frame it that weed control is being a secondary benefit because there's so many other benefits that they'll see down the line that can help just their overall production. But that secondary effect of weed control is helpful from a couple different perspectives, with resistance being one of them. So you mentioned earlier glufosinate resistance, right? And I've seen your presentations. I've seen obviously lots of weed science presentations and put it into my own, that nice table of all the commonly used herbicide groups in corn and soybean production, and how in the state of Illinois, as resistance has increased in frequency, we can keep checking off that list those available herbicides that water hemp has yet to evolve resistance to. And unfortunately, this winter, we could then finally check off glufosinate, right? So I was curious, how did that case come about? Was it one of those situations, kind of like with Dr. Hager, where a farmer had reported it, and then it got to you guys to look into it? And also, when you did receive word about this, were you, I guess hesitant on whether they were actually experiencing glufosinate resistance since it hadn't shown up yet?
Yeah. So how did these populations come to my attention? So actually, it was a combination of things. So there was at least a couple examples of just like you described. Aaron was a farmer or co-op. Somebody contacted Aaron and said, "Hey, we got a population that we suspect is not being controlled by glufosinate. We suspect it might be resistant." Um, Carla Gage at Southern Illinois University, she was a collaborator on this project. Her team there, she had a population that was not a situation like that. I think it was actually came from. She was involved with a project, I believe it was a United Soybean Board funded project to screen water hemp, just kind of to characterize resistance. And so as they were screening populations, this is one they just happened to screen. So it's kind of a sort of a quasi randomly collected population that they found. So they've come about in different ways. The second part of your question, were we hesitant? It was actually kind of funny how this started because so Aaron's group, one of his students or technicians at the time, screened the population and said, "Hey, it looks like it's resistant." And so Aaron contacted me and said, "Pat, we think we have a resistant population. You should take a look at it." Um, so I had my graduate student grow up the population to spray it in the greenhouse so that she could harvest some material from the resistant plants for collection. When she did that, she came back to me and said, "That's not resistant. It was controlled just as well as the sensitive population." And so, yes, we had a lot of skepticism about whether it was resistant or not. And so to back up a little bit, glufosinate is kind of a finicky herbicide. It's dependent on environment, particularly high temperature, high humidity are important for glufosinate to work well, and it doesn't do a very good job on large plants. And so, if you're spraying large plants, if you're getting unlucky and it's one of these really dry, low humid days out, or it's cloudy, or you spray in the evening, all those things can reduce efficacy of glufosinate. And so, you know, we've heard for years that, hey, glufosinate didn't work very well. Like, yeah, okay, it was probably not hot enough, probably wasn't humid enough. So yeah, there was definitely a lot of skepticism going into it. But at the same time, a couple years prior, Palmer amaranth had been confirmed to be resistant to glufosinate, not in Illinois, but in another state. And so we knew it was a possibility. And, kind of like what you said with checking off these herbicides, we really just knew it was a matter of time until, because water hemp has evolved resistance to every herbicide that's been thrown at it. There's no reason to suspect that it wouldn't at some point evolve resistant to glufosinate.
With that, now we can check glufosinate off that list. And so, are there any herbicides left on that list that water hemp in Illinois has not evolved resistance to?
So we don't have any resistance to the group 3 herbicides, which would be Prowl and Trefland. I don't know how many farmers are using Trefland, probably close to zero. There's a few that are using Prowl. Prowl is kind of a finicky herbicide. Those are pre-emergence products only. I guess maybe another one we could look at would be metribuzin. We do have resistance to atrazine. We have target site resistance to atrazine. We have target metabolic resistance to atrazine. Most of the resistance to atrazine is metabolic. This might confuse farmers even more. But here's a kind of a weird exception. We have target site resistance to atrazine also confers resistance to metribuzin. Metabolic resistance to atrazine, at least of what we've seen so far, does not confer resistance to metribuzin. Most of the atrazine resistance in our state in water hemp is due to metabolism. So most of our atrazine resistant water hemp populations can still be controlled by metribuzin. Okay. So we do have some metribuzin resistance in the state. So I guess, yeah, it's been checked off the list, but it remains effective on a lot of our water hemp populations. That's the only one I think I can think of that we don't have resistance to that we can use in water hemp at this point.
And I think that point goes back to what I was claiming earlier, that, it doesn't mean that every single population of water hemp has all these resistances. It's just that we know that it's possible now. So with that, how do you mentioned tank mixing. Nowadays, with this increased understanding of resistance as it relates to target site, non-target site, which you were just explaining with the complexity of that, those cross resistances, let's say, how would you, kind of encourage a producer to be good stewards of their herbicides that they're using throughout the growing season then?
Well, I tell farmers that their goal should be to not have any seeds produced by weeds in their field at the end of the growing season. So when you're managing weeds, you have a short-term goal, which is you don't want to see any yield loss this year, but you also have a long-term goal, or you should have a long-term goal of I want my seed bank, the number of seeds in my field in the soil, to be lower next year than they are this year. Okay. And so the way you do that is you don't let seeds go, or you don't let the weeds go to seed in your field. So that should be their goal. Now, how do they achieve that? And I realize they're not going to achieve that every year in every field, but make it a goal. Try to achieve that. If you can achieve that, it's going to be a lot easier to control your weeds in subsequent years, having fewer weeds. And water hemp is a good example of this. It's a good candidate for this because even though, the seed bank, those seeds that go in the soil, they can stay in that soil alive for a few years, but they don't, the proportion that are alive drops dramatically beginning after the first year. And so, after 4 years of not letting any new seeds going into the soil, your seed bank is going to go down by probably 90%. So you're going to have a very, relatively speaking, a small number of plants in your field where now you're going to be at the point where, hey, one post-emergence application is all I need, or, pre followed by one post, right? Whereas if you have some really weedy fields now, you can't probably get by with just one pre and a post. You have to do a pre and a post and maybe some layered residual and maybe another post. If you cut that seed bank back, that's where you're going to save money in the future. I know it's the future, right? Farmers are worried about making a profit this year, but if you're in it for the long haul, that's a wise investment to reduce that soil seed bank. And how do you do that? Obviously use cover crops if you can. Do a little strategic tillage. If you had a wet hole, right, where it's got flooded out, manage weeds in that wet hole. I drive by a lot of wet holes that are just nothing but water hemp going to seed in August. And that's terrible. Look, all that seed that you're producing in that field. Don't do that. Don't be that farmer. Um, so using, you can go in and just till, or even mowing it down is going to reduce a lot of that seed production in that wet area or that wet hole. So using a good herbicide program, don't skimp on rates. And I know, could be a tough year for input costs, right? But if you're in it for the long haul, skimping on rates is probably going to cost you. And then, scouting your fields, seeing what herbicides are working, changing your plan if your herbicides aren't working, and dare I say it, go out and walk through that field and get rid of those plants before they set seed. I know that's not going to be easy, but if you have a good weed control program and you're doing a good job, it's not like you're going to have thousands of weeds per acre to get rid of. You might just have a few here and there. And if you keep those from going to seed, when I see a water hemp plant going to seed, I know that plant, if it's a female plant, that thing's probably producing 100,000 seed or more. 100,000, think about that. If I take the time to kill one weed now, that'll be 100,000 less I have to kill next year. So if you think about it in that perspective, it provides some incentive for not letting that water hemp plant go to seed.
Pat, I have just one more kind of avenue for us to go down before we end here. Just briefly, the growers up here, we don't have to worry about Palmer amaranth, thankfully. But, I worked with it when I was in your lab. I know it's a big issue in, central Illinois and beyond. And you guys have been doing some really interesting research with Palmer amaranth in terms of sex determination, because like you mentioned, like water hemp, Palmer amaranth is a dioecious species where a plant is either a male or a female. And you're looking at how to determine whether plants, I guess I shouldn't even say this for you. I'll have you explain it. So what are you doing with sex determination in Palmer amaranth and why?
So we're also doing this with water hemp as well. Water hemp and Palmer amaranth are dioecious, so they produce either male flowers or female flowers. That's unlike if your farmers are familiar with red root pigweed or smooth pigweed. Those are monoecious, which means they have a single plant will have both male and female flowers. So something like smooth pigweed can self-pollinate. Water hemp and Palmer can't. Their system, it's actually been known since 1940 that they have what we say that males are the heterogametic sex. It's a big word, but basically what it means is it's like humans, whereas females are XX, males are XY. So it's the same in water hemp. So the male has two different gametes. That's where that comes from, heterogametic, an X and a Y, and it passes on either the X or the Y. If it passes on a Y, the progeny is a male. If it passes on an X, the progeny is a female. And so what we've been doing now that we have a genome is we know what that Y chromosome looks like, and we know what the genes are there. And so we can start to figure out how does this, at the gene level, how is it determined that a plant becomes a male or a female? Why the heck do we want to do this? And this is one of those things you'll read about or you'll hear about on a late night television show where they make fun of, we're doing research. Taxpayers are paying money to determine the sex of a weed. What the heck? Why? Okay, there is a reason why. It's not just because I'm sitting in an ivory tower and I want to waste taxpayers' money. There's a insect control strategy that we don't use for weeds. It's called genetic control. Okay. And the best example of that is the sterile insect technique, where they raise mass rear male or mass rear insects. This was done most famously with the screwworm. And so they'd rear insects and then they would irradiate with radiation the insects to make the males sterile. But then and then they'd release the males, they would mate with the females, but they wouldn't produce any offspring. Okay, that's a genetic control strategy. It's hugely successful. It allowed us to eradicate screwworm from the United States. Okay, could we do something like that with a weed like water hemp? So if we understood how sex was controlled, maybe we could control it. And imagine if we could make all the water hemp plants in a farmer's field males. How many seed would be produced that year? 0, right? In theory. And so if we could control sex, that could be a whole nother way of controlling these dioecious species. It's not going to work for all weeds, but for weeds like water hemp and Palmer that are dioecious, that could be a novel way of controlling these weeds. So, that's not going to happen next year. We're a ways away from doing that, but my group is looking at that. There's some other groups that are looking at it. There is a company who is actually kind of modeling the sterile insect approach, where they are testing their mass-produced palmer and water hemp pollen, and then they sterilize it, and then they release the pollen over the field, and it reduces seed production. And so that would be another example of a genetic control strategy that, like I said, to date, I'm not aware of any example of where we've actually used genetic control to manage weeds. We do it for insects. We don't do it for weeds yet, but there's a potential to do it in the future.
That's fascinating. That's really cool research. Pat, thank you so much again for joining me for the podcast episode today. I appreciate you taking the time to chat, and it's always good to catch up with you.
All right. It's great to talk to you as well, Catherine.
Once again, that was Patrick Tranel, Professor of Weed Molecular Biology and Genomics in the Department of Crop Sciences at University of Illinois. Be sure to join me for the next episode of Talking Crop, for which I'll be speaking with Dr. Allison Robertson, Extension Field Crops Pathologist with Iowa State University. That episode will release on June 10th. Until then, thanks for talking crop.